So one of the exciting things about Seymour is that it's a center, a science and technology center, and that allows scientists from all around the United States to gather together to do collaborative projects. And one example of that is, in fact, this whole cruise, and one part of that is the fact that we've been collaborating on an experiment to look at the cycling of phosphorus. So there are a lot of us that are interested in how phosphorus cycles in the ocean, but we all work on different pieces of that puzzle. And so, boy, months ago now, we started having conference calls, talking together, and trying to design some experiments to look at phosphorus cycling while we were on the cruise. We just started the third of those experiments, where we're trying to look at the bioavailability of dissolved organic phosphorus. So phosphorus comes in basically two different flavors in the water, inorganic and organic. And we know there's a lot of organic phosphorus out here, but we don't know if it's able to be utilized by the microbes. So what we did is we took water, we stripped out all of the phosphate, or the inorganic form, and left the organic form there. We added biomass back to the incubators, or the samples, and now we're trying to monitor them over time to look at whether or not the organic phosphorus compounds are being utilized.
To get this experiment started, Catherine Heal from Colorado College and Dan Rapetta from Woods Hole Oceanographic Institution developed a method to strip out the inorganic phosphorus.
We developed a method for this project that uses different types of resin for stripping different types of phosphorus from seawater. So the phosphorus stripped seawater that we're using for this, that we used with one resin that just strips the inorganic portion. And then during each time point, I also use a resin that strips the organic portion, so we can later take it back to the lab and concentrate it using existing instrumentation to try to characterize the dissolved organic phosphorus.
So it is a collaborative effort from start to finish, and each of us are measuring different parameters in these experiments, and together they'll form a full composite picture. So the OSU component is looking at biomass, so the abundance of phytoplankton and bacteria. We're also looking at nutrient concentration, so inorganic nutrients, in this case PO4, phosphate. Other groups are measuring the organic components, so that's carbon bound to phosphorus. Microbes have to degrade those organic components in order to acquire nutrients. And we're also, again, like I said, looking at biomass. We're looking at particulate carbon, nitrogen, and phosphorus composition, and how those elements change over the course of the experiment.
Sheehan Haley and Sonia Dyerman from the Woods Hole Oceanographic Institution are studying a very important enzyme.
An enzyme that breaks down organic phosphorus to inorganic phosphorus, and the enzyme is called alkaline phosphatase. So we're not only looking at whether that enzyme is present, we're also looking at the kinetics. The kinetics just means the rate, whether it's changing over time. And knowing whether it's present and how fast it changes, or if it changes at all, gives us some indication of how the organisms in this regime are making a living.
Solange Duhamel from the University of Hawaii at Manoa is making rate measurements.
Now we'll look at how fast microorganisms use inorganic and organic phosphorus compounds using radioisotopes.
I'm Mar Nietotti from the Instituto de Investigaciones Marinas in Spain, and I'm taking samples to look at the change of the composition of the dissolved organic matter over the time during the 3 incubations.
So, as you can see, Seymour is an exciting international collaborative effort where we've all gathered together in this context to study the critical macronutrient phosphate and how it cycles in marine systems. We're very excited about now working together to analyze all the data we've collected and write up the papers and hopefully really shed some new understanding on how phosphorus is cycled in this environment. We're particularly excited about comparing the data that we've collected from this station, which is very low nutrients and oligotrophic, to that very first station that we occupied where we did the same experiment, much higher nutrients and higher phosphate concentrations. So we're thinking that the organic phosphorus utilization at the first station may be very different than what we're going to observe here, and it will be an exciting comparison to look at in the future.